Chuck Drake from Johns Hopkins is going to now address that mysterious therapy that's been mentioned several times, and that is the immunotherapy for prostate cancer.
Well, I'd like to thank the SDO for inviting me here today and giving me a chance to talk about my very, very favorite subject in the world, that is the using the immune system against cancer. First of all, I'm going to talk about broadly two different kinds of immunotherapy. One is the kind you're most familiar with, the idea of vaccines or cancer vaccines that are designed to prime the immune system against cancer. I'll show you what I believe is reasonable evidence for efficacy. It was good enough for the FDA. But I'm going to talk about a little bit more provocative issues, frankly. Why is prostate cancer the first disease in which immunotherapy was approved? And what is the mechanism of action of some of these agents? And I can tell you that we really kind of don't know this. Hopefully this will just get you thinking about these subjects. Then I'm going to show you about a very different approach to immunotherapy, and that is what we call it immune checkpoint blockade. I'll show you how we think this works, some evidence for efficacy, not in prostate cancer actually, but in melanoma, and talk about how this works and how it could move forward in prostate cancer. And then finally, I'll follow up on some of Dr. Sartor's excellent comments on how we're going to integrate immunotherapy into the evolving, and I should have put evolving, prostate cancer treatment paradigm. So this is the trial that led to the first, and actually this was a landmark event for immunotherapists everywhere. This was the first vaccine trial that ever showed a clinical benefit in any solid tumor in a treatment phase. This is the IMPACT trial. It was published in the New England Journal of Medicine with Dr. Kantoff as the primary, as the first author. This trial took men with metastatic castrate-resistant prostate cancer who are either asymptomatic or minimally symptomatic. And actually, these patients could have had docetaxel or not. And that's something that's sometimes overlooked in this paradigm. They're randomized 2-to-1 to either Sipuleucel-T or a placebo, given every 3 weeks for a total of only 3 doses. And what confounded the trial a little bit and made it far less likely, actually, in my opinion, to show a survival benefit, patients who were treated with placebo were allowed to cross over to Sipuleucel-T upon progression, and about half of them did, actually. Primary endpoint was overall survival. The secondary endpoint, which was not reached, and which I won't show you the data for, but we'll talk about a little bit, was time to progression. So these are the results. It's published in the New England Journal of Medicine, about a 4-month survival benefit. You know, kind of this is what you're seeing, right? You saw this from abiraterone, right? You're seeing similar things. So the bottom line actually is about a 4-month survival benefit with a reasonable hazard ratio. And again, one thing, and to address what Marty addressed earlier, is really with very minimal side effects in this setting. What makes me really believe, though, that immunotherapy has promise both in prostate cancer and in other cancers is that's not the only trial. So there were 2 other trials of Sipuleucel-T in patients with metastatic castrate-resistant prostate cancer. Smaller trials, but showing about the same results. And this is a trial that was published in the Journal of Clinical Oncology last year as well, using a completely different vaccine, okay? This is Prostate. I'll show you how it's made later. This is a trial that was originally designed as a time to progression trial. I'm not even going to show you the schema. You know why? It was the exact same scheme as the IMPACT trial. Men with metastatic castrate-resistant disease who were asymptomatic or minimally symptomatic were randomized 2-to-1 to either Prostate or to placebo. This trial did not include a crossover, though. And it also showed evidence for survival benefit. This is obviously this is not the primary endpoint, so this is clearly a hypothesis generating, and a larger trial must be done. But bottom line, altogether, there are 4 trials in prostate cancer suggesting that vaccines have a clinical benefit. So why? I mean, vaccines have been trialed in melanoma, they've been tried in renal cell cancer and other cancer types, including even pancreatic, and really no phase 3 trials have ever been positive. So why were these trials in prostate cancer positive? Well, one idea might be maybe these are better vaccines. A lot of those earlier vaccines were peptide vaccines, sometimes not even with optimal adjuvants. Sipuleucel-T, I'll show you a little bit about how this is made. The immune cells are taken out of the body, potentially out of an immunosuppressive environment, infused back in. It's not only dendritic cells that come back in, it's a couple of other cell types. I'll show you how that works. Prostate is really kind of an advanced vaccine scheme. It uses a heterologous prime boost. It uses 2 different kinds of viruses in the prime boost phase, and also incorporates some molecules to further activate the immune system. So maybe the vaccines are better, maybe not. Arguing against that, frankly, is Prostate has been tried in pancreatic cancer, the same vaccine approach, and was a complete failure. Actually led to the company going out of business. And dendritic cell vaccines have been tried in many diseases, and they've not been successful either. So maybe it's not the vaccines. Maybe they just pick better antigens. Okay, that's possible. So PSA is maybe not just an innocent bystander. Evidence from Sam Denmead's group and others suggests that PSA has a role in prostate cancer progression. It's an enzyme. It might be involved in tumor metastases and other processes. What about prostatic acid phosphatase? The data from animals suggests that rats in particular and other primates are not tolerant to prostatic acid phosphatase. So this might be a better antigen choice than MART1 or the other antigens that have been chosen in melanoma. But maybe, and just maybe, prostate cancer is a better target, actually. So what would be evidence in that regard? So prostate cancer grows more slowly than some other tumors, particularly melanoma and renal cell cancer. All these trials actually the ones that were successful tended to pick patients who had disease that was less aggressive, actually, again, asymptomatic. And finally, this is something we always, it's all in these trials, but we never talk about. Every single patient on these trials that show a benefit were castrated, actually. And our lab and several others, including Eugene Kwan and others, have shown that androgen ablation has profound effects on the immune system. It leads in men to the regeneration of the thymus, the output of new T cells with novel specificities, and maybe that's part of the reason that we're breaking tolerance in this setting, actually. So again, this is not conclusive, but this is just something to think about going forward. So how do we think these vaccines work? And I can tell you that maybe we're actually not right here. So the idea is that the immune system starts when an activated dendritic cell takes up antigen and presents it to the immune system. And the immune cells that it presents it to are two types of immune cells, CD4 T cells and then also CD8 T cells. And the paradigm we had for a long time was you get activated dendritic cells, you get CD4 T cells, specific CD4 T cells activated, CD8 T cells activated, and these cells are going to go zoom to the tumor and they're going to mediate tumor lysis. Okay? And there is evidence for this in many, many animal models, so this is not completely unreasonable. In particular, activated CD8 T cells can traffic widely throughout the body. They can cross the blood-brain barrier even. So it's not entirely without merit. So how does Sipuleucel-T work? How does Sipuleucel-T lead to those activated dendritic cells and hopefully those activated CD8 T cells? The idea is that patients' white cells are harvested. They're cultured short-term with this proprietary cassette. The cassette includes two things. It includes the target antigen, which is prostatic acid phosphatase, coupled to GM-CSF. GM-CSF leads to the activation of those antigen-presenting cells, those dendritic cells, and to them presenting prostatic acid phosphatase on their surface to the immune system. One thing that's often not appreciated, actually, is see this little piece here? This fusion of prostatic acid phosphatate and GM-CSF, that's not in your body. That's entirely a neoantigen, actually. So the idea is that this you might have seen before, you might be tolerized to. GM-CSF is an important cytokine, but maybe this fusion represents something different to your body. The other thing is, after these cells are cultured, it's not just dendritic cells, and that's a very important point. The final infused product includes both T cells, B cells, and natural killer cells. Cells are shipped back and then infused IV. And then the idea is these are the primary cells that go into the patient. This antigen here is prostatic acid phosphatase, and the immune system is activated. Is there another way to do this? Yes. This is the vaccine called Prosvac that I told you about. It incorporates a target antigen called PSA with 3 co-stimulatory molecules to further activate the immune system. This is encoded in plasmid DNA. It's packaged into a virus. There's 2 different viruses, actually, in prime boost. One is a vaccinia virus, and the boosts are performed with fowlpox. Why is that? Guess what happens if you vaccinate with vaccinia? It works. You raise antibody against vaccinia. You can vaccinate against vaccinia really only once effectively. So the boosts must be given heterologously. Actually, Marty was part of some of the early development of this, and it was really explored nicely in phase 2 space, the idea that you have to prime with vaccinia and then boost with a fowlpox vaccine. This achieves the same thing immunologically, but a different way. So the Prosvac, the vaccine, infects either epithelial cells or directly affects dendritic cells, leads to CD4 T cell activation, and again to CD8 T cell help, and hopefully to tumor death and necrosis. Okay? Well, if that's true, right, then we should see evidence of T cell activation someplace in these patients. And the typical assay we use in the labs is called an ELISpot. Take patients' cells, T cells out of the patient, activate them in vitro with the target antigen, and see if they produce cytokines. These assays actually have not really confirmed the idea that T cells are broadly activated to these proteins. In the Sipuleucel-T data published in the New England Journal of Medicine, you can see clear L-spot evidence for reactivity to the fusion protein, but not necessarily to PAP itself, actually. In the Prostate trials, in phase 2 trials, there's some L-spot evidence, but again, really not broad evidence for T cell activation. Caveat here is that these L-spots are measuring only the peripheral response. So it could be that there's a response within the patient, within the tumor, or within the tumor-draining lymph nodes that we can't see. I'd like to raise at this point, actually, really for the first time we're starting to talk about this, the possibility of another, not necessarily alternative, but possibly complementary mechanism of action, and that is the raising of antibodies against these antigens. And remember, what is vaccinia? Vaccinia works because it raises antibodies against the target antigens. So maybe this is what happens. You have activated dendritic cells. They activate CD4 T cells. The cytokines lead to the development of plasma cells or activated B cells that make antibodies against prostatic acid phosphatase, PSA, in these different cycles. Antibodies are well known. So this is the idea is the patient's making its own version of Herceptin, right? Is there any evidence for this? Yes. I was lucky to, Nadeem Sheikh presented these data at AACR just a couple months ago, and he shared them with me. So it turns out that, in fact, the Sipuleucel-T product does raise antibodies, both to the fusion protein, which is over here, and to the native protein, which is here. So here's your controls. In your body, you've been vaccinated against tetanus, so you have IgG against tetanus. So this is a pretty high titer, about 10 to the 4th. You don't have IgM, that's the earlier version of the antibody. So those are your controls. Here's against the native PAP. At baseline, patients don't have reactivity against PAP, but after vaccination, they eventually develop reactivity to PAP. This is, you can see that the IgM is going down and the IgG is going up, right? So this is a typical antibody response. So maybe we're wrong about how these vaccines work. Maybe they work by an antibody mechanism instead or in addition to the other mechanisms. We talked a little bit about clinical issues. What are the clinical issues with Sipuleucel-T in these current cancer vaccines? One of the major issues is that really we see very few or no objective responses in patients. We also see a survival benefit without a clear evidence of time to progression. Yeah, how could that be, actually? And we can talk about this later during the question period. One thing is I showed you how long it takes to make those antibodies, right? Does it take a day? No. Does it take a week? No. I mean, you don't really see high-level IgG titers until 24 weeks later after the initial vaccination. So maybe we're just starting the timer at the wrong point. Maybe we have to start observing several months after these treatments. The other possibility is that disease kinetics are altered post-chemo. And then maybe, and one of the problems with these trials haven't really collected PSA data carefully enough, maybe we're just bending the curve in terms of PSA kinetics, actually. So again, these are ongoing issues that we don't really know the answers to. So what is another way to activate the immune system against cancer? And perhaps, a very interesting way, maybe it's entirely different, and maybe these two things can be complementary. It turns out that the T cells that infiltrate tumors express molecules that stop them from working. Okay, this is very simple. We call these molecules checkpoints. The most commonly known checkpoint is CTLA-4. CTLA-4 is an insidious molecule. What it does is it hijacks the normal signal 2 that T cells would use to get activated. CTLA-4 actually binds to the signal 2 molecules, B7-1 and B7-2, with higher affinity than the normal CD28. So this sucks up signal 2, and instead of using it to help the T cell become activated, it uses it to make the T cell become turned off. There are several checkpoint molecules that are very interesting, and they're probably non-overlapping. Some of these are probably synergistic. So we've been fortunate to participate in phase 1 trials of this checkpoint known as PD-1 with clear activity in renal cell and melanoma. Checkpoint called LAG-3, which actually shows some promise in preclinical models. So are checkpoints the same as vaccines? Actually not. They're very different. Here's how they're different. In checkpoint molecule trials, objective responses are noticed, actually. So this has been observed in kidney cancer, in melanoma, and in a series of phase 1 and 2 trials in prostate cancer. The rate of single-agent activity response is pretty good. It's in the 15 to 20% rate. And really the exciting part, actually, from an immunotherapy point of view, is that regressions tend to be durable. Some of these melanoma responses have lasted into the 5-year range. The other thing that's an important thing to note is sometimes these regressions are remarkably delayed. So 6-8 months after treatment, we see tumor regressions, both with ipilimumab and with anti-PD-1. There's a dark side, okay? And the dark side to ipilimumab is this right here. There's a 30%, actually 25 to 35% incidence of grade 3 to 4 SAEs, particularly autoimmune-related events like colitis and hypophysitis. But again, unlike the vaccines, objective tumor shrinkages have been observed. This is a patient from Jed Wolchuk's clinic with melanoma showing a beautiful tumor response. And I could show you these for prostate, renal, whatever. What about the development of these checkpoint blockade agents? Last year was, again, as was pointed out earlier, a historic time for immunotherapy and for other treatments in general. So this is the first trial ever, first randomized trial ever to show a clinical benefit in patients with metastatic melanoma. This is ipilimumab compared to a weak peptide vaccine. This is the survival curve, and a clear survival benefit was demonstrated. And as I discussed before, this was associated with grade 3 and 4 SAEs. Where is ipilimumab in prostate cancer? There are currently 2 randomized phase 3 trials ongoing. One, as Dr. Sartor pointed out, is in that post-docetaxel space. This is a placebo-controlled trial. Patients receive a low-dose radiation and are randomized, blinded actually, to either ipilimumab or placebo in a 1-to-1 ratio and followed for survival. This is an ongoing trial without really any intermittent data yet. The same exact trial, other than the radiation, is being performed in the pre-docetaxel space. That trial is a little bit earlier along at this time. So how can we integrate immunotherapy into the prostate cancer treatment paradigm? You know, 2004, Dr. Sartor showed it was very easy, right? We had androgen ablation, and then we got docetaxel, and there were some other things you could try here. This is probably an oversimplification, but bottom line, things were simple. In early 2010, in April 2010, Sipuleucel-T was approved. Most patients who are eligible for Sipuleucel-T will be in the pre-chemo space. They have to be asymptomatic or minimally symptomatic. Although, like I said earlier, it is allowed to be used in the post-docetaxel space if patients fit those criteria. Well, what happens now, actually? The bottom line, right now, in the post-docetaxel space, things are very complicated. We have 2 options we just discussed, cabazitaxel or abiraterone. What's going to happen next is this, right? So what's going to happen here? So what happens when abiraterone moves into the pre-chemo space? It's really an interesting question, and I really, really think that the right thing to do is to do randomized clinical trials, and I'm not sure it's going to be done. And this is why. Several groups, actually, Martin Cast in particular, showed that androgen ablation in animal models is more effective when given after immunotherapy, okay? My lab studied it. We actually looked at giving immunotherapy and androgen ablation at the same time or given the androgen ablation later, right? We found actually that it looked like they were probably better if you gave them at the same time. On the other hand, as we discussed this morning, abiraterone probably is going to be need to be given with some corticosteroids, at least in the current paradigm. And so what does that do to this strategy? And I can tell you this is a huge problem because we really can't model corticosteroids in mice well. Okay, so we really can't do this. I mean, the way to do this is to do a 3-arm trial with a reasonable endpoint, comparing the sequence to the combination, and probably all 3 sequences need to be combined. And again, probably this is something that's not going to happen in the future. And actually, I should. And just to point out, I mean, based on those 2 phase 3 trials, ipilimumab can also slide into those 2 spots, making everything even more complicated, actually. So what did I tell you? So I told you about Sipuleucel-T. It's a unique product. It's a cellular product. It actually includes both antigen-presenting cells, T cells, and B cells. It seems to have a really clear survival benefit, although exactly how that works is currently under investigation. Ipilimumab is in development, 2 large phase 3 trials moving along. A phase 3 trial of Prostate VF in the pre-chemo space should open at the end of this year. This is going to be a large international randomized phase 3 trial. And then finally, I really think that the new checkpoint blocking agents actually are more interesting than maybe the previous ones, particularly anti-PD-1, which in phase 1 seem to have about the same activity as ipilimumab and possibly a little bit better safety profile. This actually makes it a lot better choice maybe for potential combination trials or sequencing trials, especially in earlier stage disease, right? So with that, I think I'm done, and I probably talked too long.
Thanks, Chuck, for a terrific talk and for explaining something that not very few of us understand, and it's very hopeful that there's so many new options in immunotherapy. Now, we have time for a few questions. Yes?
Hi, Mark Exley. So either PAP or PSA, are they sufficiently expressed on the surface of either primary or metastatic prostate cancer?
Yeah, it's a great question, actually. So how much PSA, this is the Rockefeller question, how much money does a man need, right? So how much PSA do you need for it to be an antibody target? The thing that you have to remember, actually, is the presence of IgG antibodies at the site of a reactive lesion also serve to further inflammation and drive T-cell responses, right? So even if the tumor cells are dying, and that's how the antibodies are gaining access to it, it still could have a positive effect. I think the real key, honestly, is to assay these antibody titers as a function of response, right? And what I can tell you is Jed Wolchok and his group using ipilimumab in melanoma have showed really nice data that antibodies to NY-ESO-1, right, correlate with outcome, actually. So maybe this is the same thing we'll be able to see in prostate cancer.
Oliver.
Oliver Sauter, Tulane. So, Chuck, one of the interesting observations that has not been followed up very well is the activity of GM-CSF alone. And Eric Small and Doug Fig at the NCI have both indicated that you can modulate PSA in a surprising way by GM-CSF alone. I wonder if I get your comments.
That's a wonderful point, actually, and very insightful. So here's the deal, okay? So the ProstFac trial, used GM-CSF, actually. It was ProstFac plus GM-CSF. So their phase 3 trial, actually, they're going to need to answer that question. What is the role of GM-CSF? So it's going to be ProstFac plus GM-CSF, ProstFac alone. And maybe you'd like GM-CSF. Nope, it's going to be placebo, actually. So the problem is that that trial, although it will address the role of GM-CSF in a combination, not really as a single agent. GM-CSF, I can tell you immunologically, it's a double-edged sword. Too much GM-CSF, high levels of GM-CSF drive myeloid suppressor cell differentiation very, very clearly. So clearly the dose matters, and probably the location matters as well. And so I think it's a complicated issue. Really, if you had your druthers, if you were doing a mouse experiment, right, you do ProstFac plus GM-CSF, ProstFac alone, GM-CSF alone, placebo, but that's not the answer we're going to get. It's a great question.